Hi, thank you all. This is Manohar Furtado. I'm going to do a brief presentation on the SARS-CoV-2 related workflow and solutions from Thermo Fisher. We'll be talking mainly about detection systems and genotyping and a few other topics. So in the next slide, what you'll see is I'm going to start off giving a background on SARS-CoV-2, some information about how Thermo Fisher is enabling research. We'll give you an overview of diagnostic solutions, EUA approved formats, overview of some of our RUO research formats. We'll talk a little bit about SARS-CoV-2 mutations and variants and why it's important to pick them up. A couple of slides on some environmental testing. I'll end with our service and support resources. So with that, the next slide is the background. So essentially, as you all know, the SARS outbreak has had a global impact. What you see on this slide is WHO update of the global spread of the virus as of June 4th, 2020. You will see the countries, the continents listed, and the number of people who have been confirmed infected, number of deaths, and overall, this has been a very stressful solution for people all over the world. The next slide, you have a brief overview of coronaviruses. So in the red boxes here, you will see 1, 2, 3, 4 coronaviruses that do infect humans. They do not cause major disease, more like a common cold. But then in 2001, we saw the first SARS-CoV pathogenic strain come up, which caused a lot of mortality then. In 2012, we had the MERS-CoV, which is transmitted through camels, had a high mortality rate, and then you have the current pandemic with the SARS-CoV-2 in started off COVID-19. And so that's still ongoing. So these viruses have been with us for many years. As you can see the date here, the earliest discoveries translate back to 1960. They are found in domestic and wild animals, so they are among us in the populations. The life cycles for this virus is depicted in here. It's an enveloped virus, and you see the spike protein on top of it enters into the cell. It's a single-stranded, positive-stranded virus. It makes a double-stranded form of the virus. It replicates in some vesicles in here, and then ultimately migrates out through the endoplasmic reticulum, and you get new virus particles coming out here. What you see on the right is a picture of these virus vesicles within which the virus replicates, and the green spaghetti-like things that you see are the virus replicating in there. So let's go to how Thermo Fisher is enabling SARS-CoV-2 research. As you know, we are a global company. There are a number of brands that fall under Thermo Fisher, including Applied Biosystems, Fisher, and a couple of testing labs. Our mission is to enable our customers to make the world healthier, cleaner, and safer. We work on an industry-leading scale, integrated, connected reach, expanding global throughputs, and we have a global footprint. We have unmatched capabilities in terms of generating research and diagnostic products. We also have fairly comprehensive services offerings. So the next slide, you can go into that in a little more detail, where, as you'll see, we have material that we produce for protection, which is PPE, and related materials that gloves and masks that customers use. On the analysis side, we have leading genetic and cellular analysis platforms that are being used for vaccine production, gene therapy, and many other applications. What we'll be talking about today mostly is detection, which is largely using qPCR and some other technologies for viral detection and also for genotyping, as well as sequencing the virus. So that's going to be the focus today. And then we also have support for customers who produce vaccine candidates and other clinical trial logistics. We have responded in the past to many pandemics and epidemics. The earliest one was anthrax in somewhere around 2002, SARS, followed by a number of other outbreaks around the world. So we do participate in detection tests and other applications in these disease areas. In the next slide, we do have a global health program where we try to support countries that are in the low to medium income range. We have agreements with governments in many countries. We are a global health stakeholder, and as you can see, we have a single stop shopping site for a lot of products that can be used for qPCR, immunoassays, microarray sequencing, and we have a large footprint there. We focus on infectious disease, outbreaks, oncology, animal health, and environmental testing. So people have used our formats for all of these experiments. You see on the next slide is a view of our genetic testing platforms. So we do have a huge footprint in the qPCR area for real-time analysis. This is our flagship products that go on this system that can be used for detection of the virus. On the second slot here, you see that we have assays that are called allelic discrimination Pcr formats, which can be used for genotyping and variant detection. More recently, we acquired a company called Mesa, and we have a product now called Acula that is rapid testing, where you get a result within about 30 minutes. We have the ion tolerant platforms that are used for NGS extensively. We have the Sanger sequencing formats that have been around for a couple of decades, still being used. And then we have the microarray platforms for doing research on SARS-CoV-2 and other diseases. In terms of COVID testing solutions specifically, we do have EUA approval for a few kits, and the first one that got approval was the SARS-CoV-2, the COVID-19 combo kit, and the COVID-19 advanced kit. Both of these target 3 regions. So there is one assay in the ORF region, one in the S region, and one in the N region. So having multiple targets helps make the assay be more mutation resistant. So if you have mutations that prevent one of the assays from working, the other 2 are in place. We have the capability to also include a process control in there. We have multi-parametric analysis to optimize assays before we commercialize them. We do test the assays for the robustness, stability, failure modes, and then take it through EUA or CE-IVD approvals as required. So we're pretty rigorous in our assay development process. What you see in here is the intended use for the TaqPath kit and the advanced kits. So in generally, the qualitative detection assays for nucleic acids related to SARS-CoV-2, and the sample types are in bold here: nasopharyngeal, oropharyngeal, swabs, and aspirates, and BAL or bronchoalveolar lavages. All of them are approved for this particular kit, and they run on our instruments listed below. You see the QuantStudio here, the 7500s, and the QuantStudio 7. All of them can be deployed to test this assay. On the next slide, what you'll see is a general workflow. So we have an initial RNA extraction on one of our platforms referred to as the Kingfisher, and that gives you nucleic acid. The nucleic acid is then used to set up an RT reaction, a reverse transcriptase PCR reaction, which is done on our applied biosystems. So on the instruments, the data is interpreted by the COVID-19 interpretive software, and then you get a review of the controls, and then after that you get a result that tells you which of these detectors is positive, and you get a positive, negative results that's reported out. You do have the ability to get CT values, which can be obtained from the instrument, and usually the CT is an indication of the cycle number when the fluorescence crosses the threshold. in a general sense, the CT values are somewhat proportional to the amount of virus. The other instrument I mentioned here is a rapid PCR platform called the Acula. This is a point-of-care testing system. The company previously had 510K cleared products for flu A, B, RSV, and strep A. They now have an EUA for the SARS-CoV-2, which can be used. Essentially, the swabs are collected, they go into a collection media, and some of that is loaded onto the instrument, and you run it. And within about 30 minutes and 3 simple steps, you get a result. The reader is a lateral flow unit, and so you have some readout in terms of certain lines on the lateral flow instrument that correspond to the control, and a positive line for indicating presence of the virus. This will considerably improve the turnaround time and speed things up. So if I go to the next slide, then we want to talk about the other kit that we recently got an EUA approval for a couple of months ago. This is the version 2 of our combo kit. And one of the things we did here is to increase the confidence in the results. In this particular kit, we have about 8 targets within the assay. 3 of them, in 3 regions, the ORF1A, the ORF1B, and the N genes. And this compensates for any new emerging SARS virus mutations. So having degeneracy in the number of targets makes the assay extremely robust. It's very sensitive. The limit of detection is around 75 GCs per ml, so it's more sensitive than the version 1 that we had, and it's affordable and scalable and easily added onto your testing menu on the same instruments that I mentioned before. The other part where we work is in surveillance. These are RUO products, and they can be used to detect variants. So one workflow that we generally talk about is detection and identification of SARS-CoV-2 variants. So if you have a positive sample in an initial screening test, if the sample comes up positive, then it can go in for variant screening, which can be done by using genotyping assays or by amplicon-based sequencing on a Sanger instrument. You can also use the ion torrent to get complete viral characterization. So this sequence, all of the positive samples can be sequenced, and in general gives you a good result in terms of getting your entire, the clade classification. Okay. So the workflow as we see it in the field is twofold. There is one arm, which is largely diagnostic testing, where you have the qPCR assays, serology assays, point of care, and they go through regulatory requirements, CLIA cap requirements, and you get a result that goes back to the lab director and satisfies the patient needs. On the other side, we also see a need for public health pipelines where this data can go into a database and inform public health in terms of how to go about managing the epidemic, pandemic control, epidemiology data integration. And this is something we have seen with, especially in the UK with Public Health England, and more recently with the CDC here as well. There's a lot of effort in trying to ensure that you're tracking all of the variants that come through, and the methods used are qPCR, genotyping, Sanger sequencing, and other methods, and we play in all of those spaces. In terms of genomic surveillance, we do have multiple options. So one option is to just sequence the entire virus, which can be done on the ion torrent system. As I mentioned, the other option is to go through an RT-PCR step, sequence samples that have characteristic mutations of interest. So you may take a subset of what comes out of this and sequence it. And additionally, most countries would also sequence 5 to 10% of all viruses just to have a full handle on the entire mutation spectrum of variants that are floating around there. In terms of a laboratory workflow, the patient sample comes in. It could be nasopharyngeal swab, saliva, goes through a extraction system on a Kingfisher, then PCR. The results go to the lab director. You get either negatives or positives. The patient's positive, one option is to go through the genotyping format, where you can get results in terms of genotype positives. Some countries are genotyping 50 to 100% of all of the samples that they have, and this in turn can inform what needs to be sequenced. So if you find some new variants or some variant of concern or variant under investigation as defined by the WHO, those could go into sequencing. You could also have a subset of positives going directly into sequencing. So all of this is enabled in a single lab, and we have instrumentation to support all of these workflows. On the genotyping side, what you see here is a description of the genotyping assay. So what we have here in this picture in here is we have two probes, one binding to the allele 1 or the wild type, if you wish to call it that, the other binding to the mutant or allele 2, and the signal generated from these two probes is put on a cluster plot where you'll see clusters in one end corresponding to allele 1 and clusters of the other axis that correspond to allele 2. So these assays have been designed and commercialized as SNP assays. They run on the routine the PCR instruments with a slightly different software that has to be used. So this is used in screening and testing for mutations in a large number of samples. We have software that deals with this, and most of this, you'll find this on our website. So if I go to the next slide, so you'll see here are the mutation panels. What we call premier assays are the ones that are on stop. So these are the ones that are most widely used by our customer base, and they can detect all of these WHO variants listed in here. As you'll see, with time, we have more and more variants, and as they increase, we come up with assays to meet the needs for detecting all of these variants. In total, we have about 52 assays online. What you're seeing here is the ones that are most frequently used. And on the next slide, you'll see other ones that are more custom assays and used to a lesser extent, but nevertheless useful in different regions of the world for identifying what variants are in circulation. A little bit more about these variants. As you'll see, the WHO and CDC and Public Health England have lists where they classify these variants as variants of concern. And I'm not going to read through this, but it's on public websites where they have several variants that are cropping up now. Now, each of them have their own characteristics. Some of them have mutations that, due to which they can escape vaccines or not neutralized properly by vaccines. And you can see that, you can hear that more and more in the news, like especially with the Delta variant that's going around in the US right now. So with these variants, there's been a number of studies where they have shown the effect of these mutations on the ability of vaccines to neutralize these variants. So what you see in box B is neutralization of B117, and on box D is neutralization of the South African variant, the B1.351. And as you'll see, the antibodies produced in response to the mRNA 1273 vaccine from Pfizer does neutralize the B117 very well. There's only a 1.2-fold reduction, which is a very small reduction in terms of neutralization capability. However, when you go to the B1351, you do see that there's a greater reduction, about 6.4x. These antibodies are able to still neutralize many of these viruses, but reduced capability. And this is more reduced when it comes to the Delta strains. And as you'll see now, the response to this has been that very soon we probably will need booster doses to boost up our immunity. Variant testing has been going on in Europe, and there are recommendations here from the European CDC for surveillance of variants. Many countries in the EU are using our assays to try and figure out what the response is and what the spread of the virus is. And a lot of them are using this for controlling the pandemic. They do test visitors who come into the country. If they're carrying deleterious variants, they can isolate them and keep them in quarantine. So this is helping them track and test and isolate people who are being infected. A good example of this is in the UK, as you'll see on the graph on the left. The curve that you see in blue to the extreme left of this graph is the one showing the increase in Delta variants in the UK. So the UK has been using a panel of mutations from what we supply. So it's listed out here on the box on the right. So we have mutations for N501Y, the E484K, the K417N, the K417T, and more recently P681R is there because of the Delta variant. So they started off initially using 4 of the mutations, and somewhere in May they introduced the P681R to specifically pick up the Delta variant. So this is being actively used in the UK to track these variants, and the healthcare system there does use the data to intervene in terms of whatever is required for pandemic containment and isolation. So that's been going on there. And on the next slide, you have a little bit more detail on this. So the Public Health England started using this more extensively to track the rapid emergence of the Delta variants. So as you see on this slide, in the beginning, they were using largely sequencing, and somewhere around the 11th of May, they started introducing, on 29th March, they started testing the mutation panels. On May 11th, they introduced the new 681R assay specifically for the Delta variant, and they have been converting a lot of testing from NGS, though they do still continue to sequence these viruses as well. So as you see in this graph below, the dark blue bars that you see are what results they have with sequencing. This is late in May, and the light blue bars are sequencing our data got from genotyping. And as you can see, the genotyping results are available to them in about 12 to 18 hours. And so you'll see the sequencing part of it is just catching up. It takes them a week or 9 days to get sequencing results back. So using the genotyping for formats gives them faster access to what variants are circulating. In comparison, they looked at the sequencing data and the genotyping data, and as of June, what they're seeing is 99% of what's positive by genotyping is also positive by sequencing. So very high concordance between the two platforms. So they have been using this fairly extensively to be able to track down variants. What you see on the next slide is just a little bit of data in here. So if you're testing, this is data from assays looking at E484K and the deletion 6970 with genotyping assays. So if you look at it, the variant comes up in one axis of the cluster plot. The wild type is on the other axis. The software takes the fluorescence coming out of the FAM probe and the WIG probe and then plots these cluster plots where what you're actually plotting is the ratio of the fluorescence from the two dyes. So variants cluster here in blue and the wild type cluster in the bottom here on the x-axis in red. And so this can be used very effectively. What you see on the left is the data for the E484K, and on the other right side you see the data for the 6970 deletion. And these data sets are from Ola Winquist in Stockholm, Sweden, which is many European countries are using this test. And the next slide, you'll see that the reason for this variant screening is there are implications, medical implications related to the presence of SARS-CoV-2 variants. Some of them have higher capability to transmit from humans to humans. Some of them may change disease severity. Some of them may lose susceptibility to treatment like monoclonal antibodies. Some of them evade immunity conferred by vaccines. And then some of them may result in an impact on diagnostics tests if the mutations happen to be under the primers or probes for a particular test. So for these reasons, these variants are continuously tracked, and we do supply the agencies with regular updates on impact on any of our assays, as well as they're using it for tracking individuals who may harbor these variants. So complementary to genotyping is NGS and other sequencing systems. So what you see here on the left is our ION Gene Studio S5 series. So this instrument has 2 modules in there, and then you have a sample prep module and a sequencing module. So this is a little bit more labor intensive in terms of getting the library construction and other setups done. On the right, you see the new Genexus integrated sequencer. This is almost hands-off and highly automated. You can get a result in about 9 to 10 hours or so. Extensively used in biological research. For SARS-CoV-2, we can go with viral loads as low as 20 copies in the tube. We can confirm individual amplicons by Sanger sequencing if required. So this is being used extensively as well for reporting into the databases that you see here. So the genomic surveillance data with the ION SARS-CoV-2 system, it's got software that lets you plug into different databases and also view the data in reference to a reference sequence and identify SNPs and variants. The data coming out of the system can be uploaded into these different databases that you see here. It also goes into the main GCA database, into the NCBI, and also used by NextRain for global surveillance to track variants. It's a very useful platform and used worldwide in many countries. So an example of this in Japan. So there is a traveler who came in from Brazil. On day 1, he was tested at the airport. He came up negative. Few days later, he was hospitalized and tested positive for SARS-CoV-2. Within 24 hours, they were able to sequence the individual and identify the variant as a P1 variant, which was new to Japan. So that helped them implement more stringent controls to prevent additional transmission of this variant to other individuals, like quarantining this particular individual. So this is just one example of how the variant sequencing methods and the genotyping methods can be used to contain the spread of variants in the pandemic. On this slide, you see our Sanger sequencing instruments. These can be used for single amplicon sequencing. They are also used. We have a couple of collaborations in South Africa where they use this extensively for sequencing the S gene. They do provide mutation detection and can also be used for other applications. So, one other thing that Thermo Fisher has come up with more recently is something called environmental testing. And this is an instrument, as you can see on the next slide. So there is an air collection device that collects air continuously. And you can see it may be collecting air for 2 hours to 12 hours or more. The air goes in through a filter, which is in a cartridge. In the second part of the figure, you can see the cartridge in here. So this cartridge will trap all viruses, not just SARS-CoV-2. It could also trap flu A, flu B, RSV, or other viruses. The cartridge can be tested in the collection center itself, or it could be sent out and tested with one of the assays to pick up the virus. It could be sequenced as well. You can do all of it. The material is in here. You have to extract the material from the cartridge, which is a simple process, and it can go through all the workflows that I showed you earlier with a rapid testing or any other format you want, or sequencing, to get information. So the application for this is that if you have a hospital and you try to establish a COVID-free zone and you have another area that's harboring COVID patients, it's useful to have this system in certain areas to make sure that there's no COVID in circulation in the particular region of the hospital. It can also be used in industry or in schools to monitor the environment. So this is something we launched recently, and we are still working on getting all the assays to work with post-collection. So that's a really good application that would be useful in many settings. And lastly, I wanted to touch upon service and support resources within Thermo Fisher. And we have a global sales and support system. We have over 1 million customer interactions annually. You can see we have a lot of field application specialists. We have service engineers and a whole service organization that's well set up to support any needs in any country across the world. Field application specialists, it's a global scale effort and it has unmatched depth. So we do have a service program called Platinum. It's 98% uptime guarantee to get total support, meets all the pandemic requirements. We have qualification services that help meet regulatory requirements in different countries. So we will help you with some of that. Priority technical support, 365 days, 24/7, including weekends. Rapid response on site, so guaranteed engineer next business day if the instrument goes down or you have any issues of that kind. FAS training, you can have remote training with FAS consultation. They'll walk you through the workflow and the data analysis so you can get up to speed rapidly or train new technicians who come into the lab. We have comprehensive repair coverage to, you can avoid extended downtime using the support. And then we have digital remote support with on-demand tools, capabilities that can be used to help our customers. And so with that, I think I'm at the end of my presentation. So in summary, what we try to go through is that the global pandemic due to SARS obviously has infected millions of people around the world and resulted in many deaths. Additional concern has been the emergence of variants across the globe that have increased transmissibility, potentially increased disease severity, and the possibility that they may escape neutralization by antibodies. And you can see the governments reacting nowadays with suggestions of having booster doses in place. So governments, pharmaceutical companies, diagnostics, and companies have all tried to rise to the challenge to contain this pandemic by using large-scale testing, test and trace plans like the one I mentioned in the UK and in Japan, with surveillance of genetic variants. Thermo Fisher specifically enabled the use of qPCR platforms, point-of-care systems, genotyping offerings, or NGS Sanger sequencing platforms for detection and genotyping of the viruses. We also introduced antibody-based testing solutions, which I didn't talk about, but it's something else we have in our portfolio. Systems to monitor the virus in the air, which I showed a couple of slides. We also have projects for wastewater testing and testing for virus in the environment as a whole. We also work to enable sample collection and vaccine production efforts with various vaccine manufacturers and other companies. So with that, I'm going to conclude and say thank you. And for more information, please go to the Thermo Fisher website listed in here, and you'll find a lot more information on
the tests and materials available for pandemic control. So thank you, and I will end with that.